forse adesso, sì, non era collegato, perfetto. Good afternoon, everybody. Welcome back. I'm here to introduce another guest that came all the way to show us his research, and I thought that it would be a very nice topic for us from very different directions to come together on one topic. That is the one presented from Professor Guy Rosenthal. He's a very rare case of what I call brain gain instead of brain drain. No, that is very the majority of cases for Italy, but in this case we conquered Gil from the United States. He has been for a long time a professor in the United States, and in the last part of his previous career to Italy in Texas. He has worked for a very long time in close collaboration with Mexican institutions, and he has run very, again, rare and difficult types of research where you try to experiment, but in the real world, in the ecosystems, and he has put together behavior and genomics. We now, as I said, gained his knowledge and his background science in a University of Padua, where he moved 1 year and a half ago. And so it's really a big pleasure for me to listen to his talk and also to start potentially, new collaborations and new potential for us to cooperate. So thank you very much, Gail, for coming.
Thank you. Oh, thank you, Francesca, and thank you all for inviting me. This is really a unique place that you guys have here, and I visited. Francesca invited me back in 2018, and it's a real pleasure to be back. And this is, being near the Fondazione MAC and having you guys as a model was a big factor in me deciding to come back to Italy. So you're very lucky. And at the same time, I want to sort of comment on the conversation before about some of the challenges of being a PhD student, which is that since I've been moving all over the place these past few months and years, I've been talking to PhD students all over the world, and everybody is miserable. And I mean, I think that between COVID and social media, what somebody pointed out, I think that, your experience as PhD students collectively, all of your colleagues around the world, is harder and, emotionally and time-wise and work-wise more challenging than what we had to deal with in my generation. So, speaking as someone who's sort of in a later career stage, I just want you to know that, we know this, and we're here to do what we can to make things better for the younger generation that's coming up. So, all right. So we know that sexual selection plays a big role in reproduction, indirectly in ecological interactions like predation and parasitism, etc. I was just hearing about Valerio's work with using mating signals as integrative pest management on the grapes. And starting with Darwin, the Descent of Man, who elaborated this concept of sexual selection, we focused on sexual selection, and from the point of what we can easily see, what's very obvious out there, which is these extreme sexual dimorphisms, these signals that are used in sexual interactions. And Darwin had the insight that these signals evolve as a consequence of receiver decisions, as a consequence that females, usually females, prefer mates with more elaborate, more attractive signals. And we know about these signals that they're often complex and difficult to produce. They incur costs in terms of production, in terms of predation risk, and these signals are going to function differently in different environments, and they can reach intended receivers like potential mates and unintended receivers like predators. But because these signals are so obvious and easy to see and relatively easy to measure, we sort of lose focus on these receiver decisions, on what's going on in the head and sensory systems of the animals that are choosing. And not only that, but we also need to remind ourselves that choosing a mate is only one of many functions that an individual has to perform. And the same mechanisms that are involved in choosing mates, like color vision, for example, are also recruited for finding food, avoiding predators, et cetera, et cetera. So you can have this, we can think about mate choice mechanisms as having a certain contribution to fitness, and only some of this will have to do with sexual fitness, with the consequences of mating decisions. So I've sort of spent my career since I was a PhD student sort of interested in this question: how do individuals choose their mates? What are the underlying mechanisms involved? And how does mate choice shape gene flow between species? So what are the evolutionary consequences of mate choice? And more recently, I've tried to bring these questions to bear on what I think is a pretty urgent applied problem, which is really this fundamental question of whether mate choice and sexual selection are good or bad for populations adapting to environmental change. And how can we take this into account when we make conservation decisions? And I want to advertise almost exactly 1 year from now, along with my colleague at Padova, Andrea Pilastro, and Tomasz Szekely from Bath University in England. We're running a workshop in Erice, Sicily, which is a beautiful, beautiful little hill town, on sexual selection in a changing world. So there are going to be about 20 speakers from all around the world, and we're hoping for about 40 or 50 trainees. So if you're interested in this, stay tuned, and hope to see you next year. And so most of us are really interested in mate choice because we think they're likely to be agents of evolutionary change in some kind. So either by generating sexual selection on traits or patterns like assortative mating that can lead to speciation. And usually we characterize these phenotypes using behavior, like this is using T-Rex automated tracking system. And the reason we do this is because we want to start asking how these biological mechanisms, right, the stuff inside our brains and our sense organs, et cetera, bias mating decisions to form mating outcomes. And this is a study of mating outcomes by Alexa Shane and Bruce Lyon back in 2008, where they're looking at, in year to year, different predictors of mating success. And what you find is that every year there's a different set of traits that's in fashion that predicts mating success. So the question is, what do you do when these data tell us different things? Okay, so we have behavioral data that lead us to make certain predictions about how mating outcomes should work. And then if they line up with mating patterns that we observe in the wild through things like parentage analysis, then we're okay. But what do we do when these data tell us different things? And I'm going to focus on these fish that I've been working on since before I was a PhD student. I actually started working on one of these species for my undergraduate project and never stopped. And these are porta spada in Italian, Xiphophorus, and they're native to this eastern Atlantic slope of Mexico and Central America. And there's a deep history of hybridization in this group. I'm not going to talk about this too much today, but one of the things that we're finding is that hybridization between species or between lineages is a fairly pervasive feature of evolution, even in a lot of animal lineages. But I'm going to focus on is a hybridization event that happened very recently between two species of swordtails, the highland swordtail Xiphophorus malinche and the lowland or sheepshead swordtail Xiphophorus birchmani. And so these are males of the two species that you can see they look pretty different, big dorsal fin versus a pointy swordtail. And then these are hybrids with the scrambled up combinations of the parental species. And one of the things that is fun, I talked about how miserable everyone is. Now I'm going to talk about how much fun it is to be a biologist now, because we have tools that we didn't have 10, 20 years ago, where we can do a lot of really powerful stuff, even on non-model systems in the wild. And this is two former students who are both professors now, Molly Schumer and Wongfang Cui, who applied this ancestry mapping method where we can essentially paint the genomes of these hybrids to a resolution of about 10,000 base pairs, so a single gene. And we can essentially paint the genome according to ancestry. So this is the highland species, blue where it's cold, red lowland species, it's warmer. And this is a hybrid, a Malinche-like hybrid. You see lots of blue little scattered areas where it's heterozygous. And here, mostly red with these little blue chunks. So we can pinpoint on the genome where a particular stretch of DNA comes from. And we've used this for gene mapping of morphological traits, of the cancer phenotypes that the hybrids get, but we can also use it to get insights about mate choice. Now, hybridization, natural hybridization, doesn't happen in a vacuum, right? It happens in a socio-ecological context. And this context here is hang on, I'm going to get a Kleenex. I am sweating. Is it hot here or is it just me? There. The dripping sweat is not part of the show. So this is the northeasternmost tongue of the great Mesoamerican rainforest that goes down through like Costa Rica and through the Amazon on the eastern slope of the Sierra Madre Oriental here in Mexico. And hybridization is inseparable from humans. Humans are part of the story. Human behavior is part of the story. So genomic evidence and evidence from museum collections coincide and tell us that hybridization started right around 1990, which coincided with both two major hurricanes and a change in land use policy that led to deforestation and runoff and disturbance with aquatic environments. And so my research group, we've made a long-term commitment to the study system and the neighborhood where the study system is since 2005. My wife and Okay, let me go ahead and put this on a USB stick. Okay. All right. Can everybody hear me online? Okay, sorry about that. So I don't want to go through the whole thing again, but I'll give you the rushed version. I work on mate choice mechanisms and evolution in these small naturally hybridizing fish from Mexico called swordtails. And for those of you joining from home, I'm advertising this workshop that Andrea and Thomas and I are running in Erice about a year from now on sexual selection in a changing world. And all right, so these are the fish, and we can do ancestry mapping, and they live in central Mexico, and they hybridize, and here we are. So this is our research center in Calnali. It's at 1,000 meters in the Sierra Madre Oriental. We have facilities for fish and vivaria. We have a wet lab for molecular work. We would love to see more Trentini there. So if anyone wants to do tropical work. And I'm going to sort of go on a little tangent now about sort of why it's important to do this kind of work and be sort of embedded in the place and meaningfully collaborating with local people. So of course, as probably most of us know, the European conquest of the Americas was characterized not only by physical genocide, but by cultural genocide. So the Spanish saw the art, the writing, everything, every aspect of Mesoamerican civilization as being somehow connected to the devil. So they literally burned all of the codices and texts that they could get their hands on. Very little survive. And this is emblematic of the fact that Europeans came to the Americas and really didn't care about the cultural knowledge of people who had been living in these ecosystems in these places for thousands of years. And one of the things that got lost surrounded corn. And I actually wasn't going to put this in the talk, but then somebody mentioned learning how to make polenta, so I had to go there. So this is the traditional way of growing corn. It's called a milpa. So you have nitrogen-fixing beans that replenish the soil, and you've got this sustainable, relatively low productivity mixed permaculture that also provides a home for other crops like tomatoes and peppers and other things that you think of as Italian cuisine, but all came from Mexico. And in Mexico, corn is the staple of the diet all over Mesoamerica. So here, everything is about wheat and bread, and there, these are all different delicacies that you can make with corn as a base, including atole, which is a drink. Here, there's pretty much only one way that people make corn, and that's polenta. And unlike in Mesoamerica, where people had been perfectly content eating a largely corn-based diet and getting the nutrients they needed from it, when polenta was imported here, when corn was imported here to Italy, people in places like Veneto, where a huge part of the diet came from polenta, from corn, got this disease called pellagra. It's an Italian name. It's a niacin deficiency that causes eventually dementia and death. And over, from the late 1500s to the early 1900s, it's estimated about 2 million people died of malnutrition. And this was really a big part of the landscape in this part of the world. And of course, people were worried about this. And Cesare Lombroso, a famous or infamous 19th century Italian scientist, blamed corn smut, ustilago. And he thought, okay, there must be something toxic in this fungus that's killing people. But carbone del maíz is also known as huitlacoche, and it's a delicacy. It's like the truffle of Mexico. This is probably 50 euros worth of smut right there, and it actually concentrates vitamins and nutrients. It's really good for you. So it wasn't that. And I mean, it's hard to really figure out what you're dealing with if you don't even know where it comes from or what it is. So it wasn't from Turkey, and it wasn't sorghum. It was maize from the Americas. And it was actually a fairly easy fix to this problem that could have been solved if someone over 400 years had bothered to talk to a Mexican grandmother, which is you take the corn and you boil it in an alkaline solution, which removes the pericarp and releases the nutrients. It makes hominy, and this is how people in Mesoamerica eat corn. Polenta bianca, which came late to the party. So the point with this story is that, first of all, we need to think about the tropics. That's where most of our biodiversity is in the world. That's where a lot of the crops and useful plants and animals that we rely on come from. And we not only need to go there, grab stuff, and come back, but we need to actually talk to people and work with people and partner with people. And this is, again, something where Francesca, I think, is a shining exemplar of international collaboration. So let's go back to the fish. What happens when you have this kind of human interference and you have contamination from all this disturbance, hurricanes, agricultural runoff, et cetera, et cetera? how does it affect the behavior of these fish? And so one thing that you'll see is that this is true for most animals. They'll respond to species typical mating signals, and they'll usually respond to those more strongly than they will to those of other species. And when we talk about the pheromones, the urine that these fish use to attract mates, it's the same story. Females of the lowland species prefer females of the lowland species, and we just measure this by seeing how much time a female spends hanging out in one side of an aquarium with a stimulus versus the other side with the other. And same thing for Xiphophorus malinche. So you have these conspecific mating preferences. But what happens in polluted water? So if it's organic pollution, the fish are physiologically healthy, they're motivated to mate, but they can't smell the difference between conspecifics and heterospecifics anymore. And so we find this, 10 minutes? Okay. We find this in a whole bunch of different contexts. If we chemically disturb the water, if we disturb the social environment, the physical environment, these preferences go away. So we have these mechanisms, right? These chemicals interfering with olfaction that cause females to mate at random in the behavior, and then we see hybridization where we didn't before. Mechanisms, behavior, and mating outcomes all line up. It makes sense. But not so fast. Because first of all, what we see is after the initial hybridization, we see a big difference from one population to the next. You have some that are panmictic, what's called a hybrid swarm. So everyone's a hybrid and everyone's around 50% ancestry. And then you have others that are more bimodal, where we have hybrids. Everyone's still a hybrid, but you have some that are like one parent species or like the others, a bimodal distribution. And this bimodal distribution, where we've studied it closely, is stable. So you have two kinds of hybrids and very few very few crosses between these two different clusters. And we've shown that this is due to assortative mating. So if we compare mothers and their embryos, these black dots are the actual data. This is a simulated null model. And this is the difference in ancestry between the mom and the offspring. So if a female is mating with a male who's genetically different from her, it's going to have something around this region or this region. If she's mating to type with a similar male, it's going to be right here. So you have very strong assortative mating, but then one mountain range over, same situation, in fact, cleaner water in this case, you have random mating with respect to ancestry. And part of the assortative mating seems to be happening after they actually have copulation. So if we mix sperm of the two parent species and do artificial insemination, we find a very strong bias towards the conspecifics. So almost all of the fertilizations are from sperm of the female's own species. So in the reproductive tract, the sperm of conspecifics are winning. So conspecific sperm precedence, conspecific mate choice predicts strong assortative mating. All makes sense, and we can go home. But again, what we see is as soon as we take these animals from the wild, where they've been mating assortatively for 20 years, we put them into smaller spaces where they have fewer choices, assortative mating breaks down. So this is genotypes in the field, this is genotypes in the lab. And so what we should see is that there's something going on before mating that is preventing them from mating with the opposite cluster. So we should see this in behavioral trials. We should see strong preferences for their own cluster. And so we should see something like this. So parental species like males of their own species, but hybrids, though, fail to prefer their own cluster. So we don't see any within cluster preferences. And it gets worse because the genotype then doesn't predict who they like. So what does? Well, it turns out that if you break down the data by whether you collected the fish in this sunny pool or this shady pool, that is where you start getting these assortative preferences. So no matter who your genotype is here, you prefer Malinche if you're in this upper pool. In the sunny pool, you prefer Birchmani if you're in the lower pool. So genotype, the micro location, literally if you throw the net in this way or that way, predicts the preference better than the genotype. So in order to really link these, to really understand these as agents of evolutionary change, we need to understand the links between mate choice mechanisms and ultimately mating biases. So what we're working on now is trying to understand the mechanisms underlying these mating decisions and understand what promotes assortative mating and then what makes it go away. What we really want to know is how they actually do these, how they actually make these decisions in a naturalistic social environment. And one of the things we find is that social experience is key to the expression of mating preferences. For example, Birchmani prefer Birchmani if they're exposed to Birchmani. Females are exposed to Birchmani either early in life as adults, prefer their own species. They prefer heterospecifics if they're exposed to heterospecifics, and they don't show a preference if they're raised with no adults. So who you're around, who you experience, determines who you like to mate with. Oh, okay. And meanwhile, there are these species typical differences in boldness and exploratory behavior. These are genetic. These are robust to social experience. So what we're doing now is trying to understand the link between boldness, exploratory behavior, which is under ecological selection, and mate choice and as an agent of sexual selection. So my PhD student, Paola Fascinetto Zago, is working on the link between boldness, learning in a non-social context, and social context and mating outcomes. So done here, mate choice mechanisms maintain assortative mating and reproductive isolation at the same time that they can facilitate genetic exchange among lineages. And because of the link between mate choice mating behavior and boldness, they are phenotypes that, like any other trait, responds to ecological selection. And so what we're trying to understand is how this affects fitness outcomes in changing environments. And lastly, it's a little short notice, but come to our big outreach event in Mexico in May, and do come talk to me if you're interested in doing research in that part of the world. So thank you.
Thank you very much, Gail. It's one of these excellent talks that throw, a lot of questions at the end. If you had some, stability, what you thought it was the case, for sure we have more doubts and questions, which is, how our sciences should be. So before my questions, are there any questions from the audience for Gail, especially maybe from students? Yep.
Yes. Hey, Jonas, I'm a PhD student. So you talked about these two microenvironments where the females have different preferences. What happens if you take one of the females and move to the other microenvironment? Do you know? Or what would you? Oh, follow up. So can how like you showed there was like this small waterfall. Can they move between the environments, or are they like stuck? Thank you.
Thank you very much. Other questions?
First of all, thank you very much because I learned a lot and very, very fascinating story. You mentioned before that the first evidence was after two major hurricanes. So what was the change and what change that provoked the formation of the hybrid?
So it's hard to say when it was the first. There were a couple of things that happened in the late 80s, early 90s. And one of the things, one of the things, so there were these two hurricanes, Diana and Gilbert, that came through. The other thing was an economic problem. This was the time when the Mexican economy crashed, the peso crashed, and there was also a change in rural policy. So before then, people were subsidized, the government was subsidizing coffee growing in the shade. So there was an incentive to keep big trees, grow coffee under them. The coffee subsidies stopped, and a lot of people cleared their land and started ranching cattle. What all these things did, the hurricanes and the change in policy, all of them would have increased the amount of organic load, organic runoff going into natural waterways. The pollution that shuts down chemical communication is an increase in dissolved organic matter and humic acids. So this is something that doesn't harm the fish physiologically. They're still motivated to mate. Populations are still viable, but it knocks out this mechanism of communication that ensures conspecific mating.
Other questions? One.
Thank you for the interesting talk. I have a question about the hybrids. So you showed the percentage of genome shared with the two lineages, but do you also know which specific genes tend to hybridize? I mean, is it only a percentage, or is the cluster truly homogeneous as a group of hybrids, for example?
So, it's interesting, yeah. In the cluster, they're not fixed for both of the genes. So we see this ancestry variation changes a little bit from individual to individual. What we've been trying to do is understand if there are specific genes or sets of genes that are more likely to move around and are more resistant to moving around. Molly's big paper, where she showed that what really predicts it is local recombination rate. So if you have a piece of the genome that is in a recombination hotspot, so you have small pieces being transferred, they move much more easily from one species to the next in big genetic blocks. We do find, if you look at the hybrids in one river here and another river here, you find that it is predictable parts of the genome that tend to come from one species or the other species. So the pattern seems to be mirrored from one hybrid zone to the other. The one thing that we do know are going to be more resistant to movement is one of the nice things about having these replicated hybrid zones. So there are actually multiple rivers where this happens, and you can use that to identify combinations of genes that you never see in all these repeated hybrid events. And so there are about 400 genes across the genomes that are involved in pairwise incompatibilities. Like the biggest one is actually there's a respiratory protein that's half on the mitochondrion and half nuclear. And if you're homozygous, if you have two copies of the nuclear gene from one species and the mitochondrial gene from the other species, you're dead 100%. And so there are some of these genes that seem to be a barrier to gene flow. What we don't know beyond that is any generalizations about, thermal tolerance, et cetera, et cetera. None of that stuff has yielded any clear patterns.
Yeah, it's yep.
I've just seen something about the fitness of the hybrids. So the hybrids are better fitness, and the second question, or better or lower fitness. And the second question is, once the pollution in the river is restored, the hybrids tend to prefer a specific a species that is correlated with a gene set of the hybrids, or is it prefers the hybrid as well?
First of all, the hybrid males, at least a subset of the hybrid males, are more attractive to all females than the parental species. They have these new sexy combinations that give them a mating advantage. So even when the water is clean, right, I mean, this is sort of, from a conservation point of view, a little bit scary, right? You have this event of disturbance, and then even after recovery from disturbance, I mean, we go to spots that have clearly been forested since 1990 and before, and we still find hybrids because they're migrating in from more disturbed areas, and the males seem to have an advantage. Now, as far as the hybrid fitness is concerned, it's very interesting. So the one piece of evidence, first of all, that I just told you about with these genetic incompatibilities, you know that some of these hybrid combinations, like some hybrids, the ones that have mitochondrion here, homozygous there, dead. So that's clearly bad. But on the other hand, if we actually look at the phenotypes of hybrids in the first and second generation and compare them to the parentals, so of course we're losing the embryos that die from these mitochondrial incompatibilities. We never see those. If we compare juveniles and adults from birth to adulthood, then we see no fitness effect of hybridization in the first couple generations. But It's very interesting because we tend to think of, like, in the hybridization literature and genetics, people talk about intrinsic incompatibilities and extrinsic incompatibilities. So if you've got something that screws up your cellular respiration, like this mitonuclear thing, it's intrinsic. If you've got something where you're too cold adapted for an intermediate temperature, then that's extrinsic, that's ecological selection. And something happened that makes me not believe in that dichotomy, which is when we first started studying these hybrids and starting to make these crosses in aquariums indoors, it was very difficult. It took us a long time. We got the first generation hybrids. They looked like hybrids. They were fine. Then we tried to cross them to each other and make second generation hybrids. And we got 97% mortality before the first 3 days of life, 100% after a week. And so we thought, okay, this whole hybrid story has to be because of backcrossing, right? Hybrids are mating with the parentals because they can't mate with each other. Meanwhile, in Mexico, we had these big mesocosm outdoor tanks where they're getting sunlight, where they're getting natural food, etc. And we'd left these F1 hybrids alone. And again, we have these second generation hybrids that we can't tell a difference between them and the parentals in terms of fitness. So something about that environmental change from the lab to the semi-natural mesocosms, we think it was probably the broad spectrum of light that was the key factor, went from 100% mortality relative to the parentals to 0. So there's some really interesting stuff to be worked out about how environment works out with these hybrid combinations.
So I have a question then. So do you think that the main hypothesis is gene expression then, more than actual genome per se?
Oh, thanks. In terms of the hybrid dysfunction, it's hard to say. So Molly was doing some work on trying to look for allele-specific expression. So whether in the hybrids, they're more likely to express an allele from one parental species or the other. And I think for technical reasons, they didn't get very far. One thing you find with hybridization in general, and we haven't really looked at this in this species, but people have found in a lot of, is that even when you don't detect phenotypes, you detect these big differences in gene expression that suggests some misregulation is happening, but to some extent it seems to get buffered by these other processes. We've done a few gene expression studies with behavior, and you get these big grocery lists of differentially expressed genes. I don't have too good an insight onto that.
Yep, that's Mario.
What is going to happen if you remove the hormones in the artificial environment? Are they still mating? Or, yeah, I mean the signals of the hormones. Pheromones.
They definitely still mate. I mean, they definitely mate in polluted, that's the thing. So they mate in polluted water. Thank you. So even though they're not perceiving the signals, they're still mating. They're just mating at random with respect to species. No. So that's how you get the hybrids, is because they're mating, but they can't tell who they're mating with.
Okay, I would have more questions, but thanks again, Gil.
Okay, so we go on with the round table of the biodiversity, ecology, and environment. We have three students from Zoom participate in this round table, and David Andreatta, who is here, and the moderator is Dr. Francesca Cagnati. Yes. Able to put them up. Yes, Francesca. Sì. Francesca, ciao. Can you hear me? Francesca?
Francesca, ci senti? Yes,
Sì.
it's really there.
Can you hear me?
Yes, very well. Thank you, Francesca.
So, Giannovelli Lucia, are you?
Yeah, there we go. Here she is.
And Talia Giulia.
Hello.
Okay.
Okay, perfect.
So
Hello.
let's start.
Yes. Okay. So thank you very much for connecting and to be my, in-person supporter here. Actually, it would be nice if, yeah, I don't know where the camera is, but so we need to coordinate a bit, although we are in hybrid mode. And I thought that many questions were asked before. You may have followed remotely. And Valerio actually took one of my questions, which was, what would you see of unique in your research that you would really consider your breakthrough? But now I have remodulated the question, and I think it's nice to discuss about one of the elements that emerged before. So the fact that we are pushed to hyper specialization. So for sure, in your PhD, you become very good at one thing. And allegedly, the moment in which you defend your PhD is very different from when you defended exams or even maybe a master's, because you are becoming very knowledgeable and, hopefully even more than your advisor in that specific topic. But at the same time, I think, and this is maybe a personal take, that it's still very important to have a synthetic mind. So to be able to put things in context and to look ahead in terms of challenges and creativity, especially because of the big challenges that we are faced with, both environmental and societal. And so I would like to hear from you about this. So if you had to say how your specific research can be put in perspective of either purely scientific or societal challenges, what would you say? And why not even in an outreach mode, because it is important to make ourselves understood by the rest of the community, scientific community, but also from the public. So I don't know how we want to start. We start from the local, and then we give the word to those who are connected remotely. Please, David.
Hi, good afternoon. And I work in the field of grassland remote sensing. To answer this question, I think I would tell you about my background, because I think that having a different background from remote sensing specialists helped me to understand the background of the topic. So maybe the initial project was very ecological about the shift in phenology in the recent decades. But then looking at the phenology, we understood that looking at that time series, we could map mowing frequency, for example. So the fact that I come from agronomy field helped me to understand that that's important for the agronomy field, for the conservation policy, for management action and policies. And so from that idea, which we switched to the idea of mapping mowing frequency, and then we are back to phenology. But having in mind both, so the high science and maybe a more technical, and both practical use of scientific products can help us in having good ideas which are interesting both for science and both for the society, which is also important.
Yeah, I really like this. So basically, having a different background really helped you think out of the box in essence, and to really see to have basically an external point of view that actually conducted you to a solution of what you were observing. Yes, and by the way, I didn't ask you explicitly to introduce, but you introduced yourself, so I will ask the same to you guys connected remotely to maybe give your feedback and at the same time introduce yourself. So I don't know who would like to come next. I don't know, Giulia, Francesca.
I can start if you want.
Yes, please, thank you.
Okay, so my name is Francesca, and I'm about to submit my thesis. So in my PhD project, I applied a multi-omics approach to, on one side, to reconstructing the impact of natural and human-mediated activities on alpine chamois. And in the other part of my project, well, in this part I use the genomics, but in the other part of my project I use the metabarcoding to study the diet of chamois and to investigate if and how overlap with the red deer and the domestic sheep can induce changes in chamois diet. And to answer your question, I will talk about the third part of my project, that is the interdisciplinary part. Indeed, in this part, I performed an economic analysis to evaluate the trade-offs of several intervention measures that aim at reducing the potential competition between those species. So I used this part to answer your question because I think that this project gave me the opportunity to have a broader view of the problem and to put in practice what I studied from a scientific point of view. So I think that is important to yes to have a good knowledge of the scientific part, but also to collaborate with the other fields. So, for example, in my economic analysis, I also considered the socio-cultural aspect besides the economical ones and the environmental ones. So for me, it was really useful also to not only to understand this socio-cultural part of the problem, but also to gain other skills in something that I was not used to work with. So.
Yeah, no, this is also very interesting point, also to not only find new knowledge yourself, but also trying to connect with other expertise and enter into, I guess, also different, it's always difficult to communicate. It's a different language, it's different categories,
different mindset,
exactly. Different mindset, but you need them to communicate to really push the knowledge forward, especially in Anthropocene, as also Gil's talk showed. If he hadn't asked himself, what has happened to this river in the last 20 years that might have changed things like pollution, the hurricane, he wouldn't get to, like basic understanding of genomics as he did. So it's basically the same approach to really try and ask questions beyond the first level, the first objective. And my question, maybe if I can follow up, is did you have this in mind from the very beginning, or is something that you developed throughout your PhD course as well?
No, my PhD course was interdisciplinary, so I know that I had to develop the interdisciplinary part, but the focus of the interdisciplinary part, I developed it during my PhD process. So I had to make a literature review to understand what are the main problems, and after that I decided to develop this part in this way. So yes, I think it was useful also because sometimes when talking about wildlife conservation, sometimes you don't consider some aspects, no? So how people will accept this measure or what is the economic impact. So I think it's important also to increase knowledge in local people and to increase also the like the conservation part in people. I don't know.
So now that's and somehow, you know, it pushes you to get a more comprehensive story, although maybe it's not the cleanest story that you would love to have to publish, no, but you have to accept complexity, which means taking into account the socio-ecological part. Very interesting. Thank you very much, Francesca. Maybe, I don't know, Lucia, Julia, who would like to go next?
If it's okay, I
Yes,
will
of course.
go
Thank you.
because-
Remember
okay.
to introduce yourself as well. Thank you.
Yes. Okay. So I'm Lucia Zanovello, and I work on amphibian conservation. And similarly to Francesca, I also am inside this interdisciplinary course. So I also have another subject, let's say. And so during my PhD, I basically developed and applied monitoring protocols based on environmental DNA. And so with them, I collected data not only on the presence of amphibians, but also their genetic diversity. And the aim of my project, okay, there was a moment I didn't see you, okay. The aim of my project is to basically combine this genetic data with also ecological indexes and also indexes that are derived from landscape planning. Because I'm interested in studying not only natural habitats, but also artificial or otherwise human impacted areas, and to try to understand how amphibians are actually using these kind of habitats right now, because they could be important also for future conservation planning. And so, yeah, for me, it has also been quite a challenge to try to have a broader view of the project and to put together expertise from especially the area of landscape planning, because I'm collaborating with an architect. So you can imagine that it's quite a different field. And but it has been a challenge, but I feel that my project in the end is more rich with this kind of approach. And I still have to see the results because I'm in the third year now, but yeah, I think it's been interesting anyway.
And what was I mean, I can imagine that that must have been quite difficult, because again, the categories, you know, the mind categories, the mindset is very different when you mention, you know, like landscape for us is a matter of habitat and biodiversity. For an architect is basically the structure, and it's sort of, you know, a bit conceived like a design, you know. So what was the most difficult component? I mean, really explaining each other, a matter of language, a matter of goals. So what was the most challenging aspect?
Yes, I feel it was both a problem of terminology, let's say, because as you said, maybe landscape for us is one thing, more like from a natural point of view. And also, since I also wanted to consider also ecological indexes, so I had already some defining features for my habitats, and then I had to put together also those other indexes. And for them, maybe it could be like presence of buildings or the road network. So it was more from a human point of view, let's say. And I feel maybe the most difficult part was at the very beginning, in which we had to decide how the two fields could actually work together, because I started that I had an idea about what I wanted to do from the biological point of view, and a very general idea about how to make everything work, let's say. And so I think I have been lucky with the professor with which I'm collaborating, because he was a very, how can I say, very eager to work with me. Yes. And so we had to discuss quite a bit, but in the end we were able to identify which indexes from their field could also be applied in my case. So yes.
Yeah. No, very, very interesting again. Yeah. And Julia, last but not least.
Yeah, thanks. And so, hi everybody, I'm Giulia Calia, and I have an agronomy background from the high schools, but then I became a computational biologist. And in my PhD project, I'm focusing on the characterization of the genome, firstly of the genome of these plant pathogenic bacteria that is phytoplasma and infects apple trees. And then to study which are the genetic factors that are behind their pathogenicity and their invasivity. And with a focus on that, I'm currently on my period abroad of the PhD, and I'm finalizing the development of this machine learning algorithm for protein prediction, because we know that this pathogen is encoding for proteins that are mainly involved in the development of the disease. And we know very few about these proteins, and the current methods for protein prediction are becoming unreliable, let's say. And so developing an efficient predictor for these proteins can help us to gain knowledge about the molecular basis of the disease, the interaction between pathogen and host plants, etc. And so to answer to your question, basically, yes, we are developing hyper-specific skills, let's say. But for computational biologists and bioinformatics in general, I think that we can use the tools that we have, like machine learning, algorithm, etc., to a variety of application. So the methodology is basically the same, and then the data are changing. So in this sense, we gain plasticity because we have to deal with different type of data, but then the methodology that we can apply are basically the same, and we also can think about how to apply all this stuff to different fields and to different topics, let's say.
Yeah, no, it's a very interesting point, this one, no? The plasticity that helps you to first of all take a new problem, analyze it, and then find a good match with methodologies or approaches. And can you maybe tell us a little bit about your experience, you know, but like a key moment of your research where there was a key question that you had to basically translate into a methodological approach, or maybe learn a new methodological approach, or maybe even a moment of difficulty where
Yeah, no, yeah, there was basically at the beginning of the second year, we started to think how we can predict these proteins and how we can classify and distinguish this protein between among all the other proteins that the pathogen is encoding for. And so yeah, I had to learn different methodologies. I mean, I already learned that from my master's, but then learning and applying them is a different thing. So just we have to decide in which way to move, if moving through the machine learning world, or just developing a script that can discriminate between some characteristic of the protein, etc. So yeah, the big game changer, I think that it was this one. So to decide where to go, and then what to apply, and see if your decision is the right decision or not.
Yeah, thank you very much. This is also very
Thanks
nice. And I'm asking maybe second and possibly also last question that is a bit broader. Before we noticed how much this generation, your generation, this generation of students, of PhD students that are aiming at a research career, whether academic or applied, have maybe a lot of possibilities with respect to what used to be, but at the same time, a lot more challenges, right? And what really touches me, I mean, it really strikes me, is the great ability of this generation to be very, like plastic and fast to change. Like change is really embedded in your generation somehow, because it happens so fast. And so maybe a bit of a provocative question is, what is a term that we use a lot in this sense is resilience, but it's also a term that is very important when you look into conservation and into the current challenges. So what is your own definition, your own take of this term, both personally and also maybe from a broader point of view, from from the point of view of ecosystems and conservation, or new pathogens and challenges. And I would like to open up possibly also to others in the audience, because I think it's a very challenging question from everybody. So we start the turnaround again.
Okay. Okay, speaking about resilience, two main themes came in my mind, and one is more personal, which is going abroad, which was a bit challenging in my mind, but at the end was really very fascinating. I really think it changed my career, but also my mindset, and so it was a great experience. So from one possible disturbance moment to really growth moment. And then, yes, I would think about the start of the PhD as a moment where you have to replan everything and to understand new methodologies, to start programming. And this is quite interesting. And the resilience we see in the ecosystem is one topic that we are discussing in these days, to understand which are changing, which are not, and why are some ecosystems so resilient to changes. And I think this is quite fascinating. In some cases, it can give us some hope about the future.
Thank you very much. And others from the audience, I don't know, would you like to use the same order or any other order? Francesca, would you like to? Yeah.
Well, personally, also for me, it was because my PhD was first 2 years in Trento, then Madrid, then Ferrara. So for me, there were a lot of changes. And also for me, it was like adapting myself to different methodologies, different arguments. So for me, was the PhD was challenging in this sense. And resilience for me was also, when you are doing something and it doesn't work, but then you say, okay, I need to try again and again. And sometimes you just have to change your point of view, or you just have to change the methodology. And at the end, your initial plan changed, but you can anyway reach your objective. So
yeah,
this was for me.
Yeah, thank you, Francesca. Lucia?
Well, okay, I can go then. Okay, so I feel that the PhD in general is a period in which you have to be very flexible and to adapt to how even your project itself changes, because usually you start with an idea and then you try to develop it, and then usually it broadens a lot. So then you have to decide which part is more interesting maybe, and which part can be another project in the future, I don't know. So I feel that it's a continuous challenge, that you have to keep your mind open and to even change your idea as you go on with the project. And yes, as I said before, having an interdisciplinary project was another challenge in that sense, because I had to work with people from different areas, and yes. And also inside my project, I'm interested in this idea of changing environments with the impact of humans, but also with climate changes, I mean. Because studying alpine amphibians, I have the opportunity of having habitats that are maybe across different elevation points. So I can see if there are differences between amphibians living in the plains or at the upper sites on the mountains. And this has been also a challenge in the last few years because I saw that, for example, sampling has become more and more difficult due to the climate changes because the season is completely modified right now with higher temperature earlier and also maybe drier springs. So for me, it's been difficult even to find my target species sometimes. So yes, it's a continuous challenge, but at least you are, I mean, you have to keep thinking about the new ideas and maybe change the direction in the project. So
yeah,
yes.
And at the same time, you were really exposed to what could mean to be resilient to abiotic changes, you know, from species point of view, because it's really like a habitat becoming shrunk or varying. And finally, Julia.
Yeah, I don't know what else to add to guys, but yeah, I think that the main keywords that can be associated with resilience is learning, because without learning, basically you're stuck in the same errors, in the same position, and so yeah, I think that the only way to go on and to adapt is by learning yourself from your errors and from also from the challenge that's exposed to you, you know. Yeah. So yeah, only that.
Yeah, no, I think really key. Never assume, but try to learn and ask new questions.
Exactly.
Yeah, that's a very nice conclusion, I think.
Thanks.
Okay, so thank you very much, guys, for your availability. Really great conversation.
Thank you.
And thanks again to Gil for coming.
Thank you, Francesca. Thank you. I would like to call now to the stage Dr. Franco Biasoli, who will be the moderator for the next roundtable, and the PhD students Leonardo Menghi, Maestrello Valentina, Michele Ricci, Rosalba Roccatello, and Giorgia Secchi. For the other group discussion of the area food and nutrition. So students are invited to have a very short talk about the topic and give your name, please. And then you start with the group discussion. Thank you.
So, good evening, everyone. My name is Michele Ricci. I'm a third year PhD student. I have submitted my thesis and I'm about to defend it in like 3 weeks. All my project was about data analysis applied on quality control procedures of food, and the whole project focused on developing models to improve quality control on a real case scenario. 2 works focused on the developing of a statistical model to infer the effect of specific conditions of the project on the final properties of food products, and other 2 activities focused on the developing on predictive models. Results brought to 2 articles and many conference proceedings. And I will pass to Rosalba.
Good evening, everyone. My name is Rosalba Roccatello. I'm a third-year PhD student here at Foundation Edmund Mack, and my project is focused on the use of insect in animal feed and the approach that the Italian consumers has with this new kind of products, especially poultry and fish, so animal origin products from animals fed with insect-based feed. And basically, the 2 main aims of my PhD project are to collect data on consumer behavior approach, so about their willingness to accept, to taste, and to pay for this particular product, and also to combine this data with the sensory data analysis. So I also try to understand from a sensory point of view what is the approach of Italian consumer, especially with the poultry products fed with insect-based feed, and we found some interesting results about this novel food, and yes, basically, this is my PhD project. I pass to Leonardo.
Hi everyone, my name is Leonardo and I'm a PhD candidate and I will be defending my PhD thesis in 3 weeks as Michele. And my main topic is about sensory perception. So I was particularly interested in assessing why people do taste or smell in a different way. And so we were quite interested in the novel factors that are supposed to affect sensory perception, which is the microbiota. And so we collected a very huge amount of data about smells, about taste. So people tasted lots of very nice food products, I would say. And we collected data about saliva and feces. And so we try basically to merge all those data to explain additionally whether sensory perception could or not be affected by the composition of our microbiome. And also we collected data about diet. So we got very interesting results, and it was a very long journey, but a very nice journey as well. So I will be passed the mic.
Hi everyone, I'm Giorgia. I defended my thesis of PhD 2 months ago, so very recently. During my PhD, I'm working on applying the metagenomic approach to rumen in testing and milk in relation to environment and quality of products, and we consider the summer alpine transhumans as a case study. So basically, we investigated the differences and the changes before, during, and after summer transhumans of dairy cows. So we decided to apply this approach because there isn't anything in literature that combine together the microbial data and the physical-chemical analysis. So we still found in some cases strongly interaction between these two type of data, and other cases no. This argument is very interesting for me, and we want to keep in deep on this knowledge. Thank you.
I'm Valentina, and I'm a PhD student at the 3rd year, and I work in the traceability unit. So my PhD project is about the authenticity and traceability of food. And in particular, I developed new analytical methods based on the NMR spectroscopy to assess the authenticity and traceability of Grana Padano cheese. And in particular, I dealt with the problems of aging, authenticity of Grana Padano against its main competitors, foreign competitors, and I try to detect if it's possible to see in the cheese the different productions. So there are two ways to produce Grana Padano cheese, one the traditional one and a new one that the consortium is going to analyze and study. So I try to see if in the real cheese there are differences that could be seen with the NMR, so in the metabolome, and the answer is yes. So my PhD is divided in 2 parts, the analytical part with the NMR and the statistical part. So I dealt with the huge amount of data, and I built a statistical model to detect the authenticity.
Well, thank you very much. Now I have to lead the discussion. Maybe I would start when I read the title of the presentation of today, the keynote presentation. I was a little bit kind of upset because we are in a place where we talk your language more or less. We have sensory, we have Grana Padano, we have cheese, we have NMR for traceability, authenticity, and all this kind of stuff that were not represented. Although, for instance, I learned a lot about Phylogomyiasis, which I considered a pest. Now I think can be something more interesting, probably. But so on one side I was accepted, on the other side it was very interesting because it's better if you listen to topics that you don't know, so you learn a lot. And can you comment on your impression on this? So how do you think that your particular narrow, I would say, point of view related to sensory and gut microbiota or cheese and cows and whatever can be linked with a more global view that we got today from the topics that are, because we learned, for instance, again from the last topic, that if you change from coffee to cows, then these poor fishes that do not know anymore what they have to do and so on. So there is a link on everything. And can you imagine how your activity can be linked with a broader stuff on the other side?
Okay. So I took the opportunity to listen to very interesting topics and researchers speaking today. So what I can imagine is to usually during the PhD period, we are focused on our project and we think just to our small project and just to small problems that we have to face day by day. But maybe listen to other researchers and in other topics and in other fields, we could enlarge our research and maybe try to find different links. For example, in my case, it's a bit difficult because I'm a chemist, so almost none of the speakers was speaking about chemistry and so on. But for example, the second speaker spoke about isotopes. So maybe not for, even for me, but even for my unit could be interesting to that they work on isotopes. It could be interesting to even enlarge their field of application because they work mainly with foods yes, with foods. So maybe listen to others and other groups. You can expand your knowledge and even expand your research and applications.
In my opinion, all the topics are very interesting, and in my case, for example, I applied the metagenomic on the summer alpine transmass, but I thought that it will be interesting for this, for example, for in terms of traceability, for enhancing the traditional products, for example. But also it could be interesting expand this type of study in another part of the world. So to find a different relationship, different behavior. So it's very interesting.
I think in my case it's quite easy to answer because I work with people. So basically we can easily connect each kind of topic with people in general. So, but according to the topics that has been addressed today, I would say that's the very fascinating mate selection topic is, in my opinion, can match overall 100%. Just think about that. One of the first paper linking sensory perception with microbiome was on Drosophila. So basically, actually, who knows whether using, for example, germ-free mice versus mice with a specific microbiome could or not affect, the mating between mice from the same cohort. I don't know. I'm not expert at all about this, but I find it very fascinating.
I agree with Leonardo because I also work with people and in consumer behavior studies, I mean, the interdisciplinarity is a keyword for our study because especially during my PhD, I had the opportunity to face with behavioral economics, consumer behavior, sensory sciences. So a lot of topics that match with my my topic of the PhD project. Also, I would say that studying insect use as food and feed is quite challenging because during my PhD project, I touched a lot of topics, and when the second speaker, Professor Rulli, spoke about food insecurity, so yes, food insecurity, especially in my case, insects are considered one of the sources that in the future could be used to solve this problem, not at all, but could be a chance in this way. So I think that for me, in the interdisciplinarity is fundamental to reach my goals, and so basically this.
Personally, I would love to say that I agree with Leonardo, but unfortunately I arrived late at this event, so I did not attend it at previous conferences. Fortunately, I attended the last one, which was very interesting. But I can answer saying that attending at many presentations, many conferences on about the field of data analysis was very useful, a lot of time fundamental to improve work, because for specific and complex problems, it could be very useful to see a specific solution that had been developed in certain cases. Many choices that I took for my paper came from presentation and conferences that I have attended online or in person during my PhD. And that was the first step. Then I've always tried to contact them to ask more, but the first step, it's always to have a chance to know, to notice the existence of different approaches, and even in a field like statistical analysis, which seem very automatized, but needs to see all the new and different solutions available.
Thank you. Obviously, I have maybe one or two questions more, but if someone would like to ask general or technical questions to the PhD candidates, okay.
Waken up by that. How are you using the isotopes in your kind of project? I mean, do you mean natural isotope, naturally occurring isotopes in the samples, or do you treat them with some molecule containing highly enriched in isotopes, or how do you carry out this kind of analysis in your kind of samples?
I don't do analysis on isotopes. My group do the part. My project was to develop an NMR method to support the models that they built on isotopes. But I can tell you about isotopes anyway. So isotopes, hydrogen, carbon, oxygen of the real cheese, of the actual cheese. I don't know if they extract in somehow, for example, for wines, they extract the ethanol, they ferment the mast, or from the wine, they take the ethanol and they analyze the ethanol. On cheese, I don't know how they do this, maybe on the bulk, but I'm not sure about that. Anyway, carbon, hydrogen, and oxygen. They are the official parameters for authenticity of cheese, of Grana Padano. So, and my project, yes, is to support these models with the NMR, with the metabolomics part, but with NMR, basic NMR.
I didn't know, and this is why these days are useful, that there was a study in FEM about the transhumanza and the microbiota associated with that. I did the same with migrating ungulates, which is probably what our ancestors learned this from. No, they saw that the deer would migrate up and down the mountains and said, oh, maybe we should do the same with our cows. So I was really interested to see how integrating a social component like the traditional transumanza could really explain maybe a lot about the richness of the environment. And so maybe did you see some interesting aspects about this, like especially linked to what could be a contribution of biodiversity from this social behavior of taking animals up and down? So you said that sometimes it worked, sometimes it didn't, but
the rumen intestine products and milk, but for the moment we didn't analyze the social part. So we basically see the for example, for a small batch of sample that from 4 different summer farms in the same area, we also saw very different microbiome, and that they change very right. Right. Yeah. Basically, this changes is not from only a single factor, but could be a different biodiversity of pastures, different management, the adaptation of animal on the summer pastures. So this argument is very interesting in my personal opinion, and I want to go in deep for technology. Also to applying this approach also in different other species. For example, I like very much the goat, so I hope in the future applying this also in the goat species.
You can suggest literature on deers, or you can suggest something on cows and goats. Other questions from the audience? Or so I think we are probably willing also to close. I have many questions, but maybe the last one is the easiest one. Do you have suggestions for the institutions that were supporting your adventure here, and for your colleagues that are following you now for the first and second year? I wonder, for instance, if you can tell something about this round table, these things, and in general. Diamo di vedere là, quello da qui, a partire da Leonardo e poi.
Okay, this is a tough question, I would say. I know. Okay, so for my colleague, the first suggestion is to go abroad. To go abroad and try to experience, to know as many people as you can, and to try to observe everything from other people. And the second suggestion could be as curious as you can, in a sense that you should try to know several different topics in the same time. So just before we were talking about being highly specific in a topic. According to my experience, actually, the success, I would say, of my PhD was to know a little bit of a lot of arguments. So this is a kind of suggestion I would like to give. And about institution, about today, probably there should have been, a topic more, as you were saying before, like a topic that was supposed to cover a bit more our specific topic, so the food, but basically, and probably also in the offer of the PhD courses, probably just to think about how many different topics are addressed by the PhD program. So because in some cases, especially in the first year of my PhD, there was somehow a kind of polarization of topics towards, ecology or similar topics. But from a food point of view, there were no so many PhD courses. Okay, so those are my suggestions.
So, as I said before, the key of my PhD project is interdisciplinarity. So the suggestion that comes from me both for colleagues and for a senior researcher is to develop the interconnection between groups, between people, because, for example, I have a lot of ideas for my PhD project, but sometimes it's quite difficult to develop because, I mean, I'm just a PhD student, so I have some experience in some topics, but maybe sometimes it could be useful to have some hints from seniors, maybe also from other groups from totally different topic. And I think this is the key to help us to create and develop projects that are very interdisciplinary, and also for the development of research, because from a big project, anyone involved could take something for its research. Thank you.
I guess that very important topics have been said already by Rosalba and Leonardo. So I will say to first year PhD students, talk each other and be curious, especially on all the possibilities available here and abroad.
I'm agree with my colleagues, and I also give another suggestion. Also, thinking about the future applying of the research, so the future perspective and the future development, improvement on all the topic.
Okay, I agree with everyone, and even maybe. Maybe have more occasions like this, maybe in a smaller way, just between PhD students, because when we are just between us, among us, we are more free to ask questions between us. So I can ask maybe in a more strict way, for example, to her, how did you do this? But in a technical way, practically, not just theory. And so maybe in another group, there is another student that is doing the same thing as I did, but so we can exchange more freely. That's the suggestion. And improve the part courses about chemistry.
Okay, so I think there are obviously many other topics, but I would suggest to stop here, and I should give the microphone, I assume, to the director.
Just to finish, I don't know if there is any. Gerardo is not here, Riccardo is not here. Also on behalf of the University of Trento CTRA, I think it's important that we have more often this moment, and especially with the comments that you have been giving in this particular show, special show that you do it at the end, and we don't give enough opportunity to you for discussing, for interacting, to move out from the research unit you are and attend many other occasions, other possibility to hear science. What is important in the high formation in PhD, when you go to, important university like Berkeley, we have been listening for Berkeley, they are professor in Berkeley. So if you attend a program, PhD program in that university, you really have a special moment for what is called lateral thinking. It's not, you do your science. Your science, you can discuss your science with your supervisor, and then you need to discuss with your community, because then you can understand and you can share many opportunities, the opportunity to meet other people, to join other people, to understand what is the feelings of the other people, what you can learn from the others. First of all, you are learning. We are all learning here. There is no one that knows everything.
So we are all learning. So, and learning is the most important. And formation in learning, information in studying, it's all about PhD. So it's the first moment that you have for starting your own career, but it's important for the research unit you are, because you are the interaction person with all the others, the research unit inside the center, but also with all over the world. So I think it's important to have more opportunities, more chance to meet each other, not only once, but we have to find the way. And I just this morning is stimulating to say you can decide it, for instance, seminars that are please out of the bin. So out of the bin, think lateral and invite people. We will find a way to bring these people here and discuss and having more occasion to days like this. Thank you very much for the last but not the least, I would like very much to thank Elisabetta Perini. Please come here. Elisabetta is the welcome office, is the person that every day is asking me, we have this other student coming in, please please sign the entrance of this other student. So without her, all that happened today would never happen. So please remember that she's the person to whom to be in touch, but also the person that is also interacting with me to know exactly what is the sentiment for the for the high formation and PhD courses. Grazie. Grazie. So the appointment is next year, but I hope it's not next year. Not.
Adesso cerco di togliere senza danno